Let me just welcome everybody to our April Plant Phenomics Fridays from Satellite to Seeds Spring Seminar Series. I hope many of you like alliteration. We're meeting here at Iowa State and also online. This is our first in the series for April. We figured if we put it April 1st as the first time we meet, that could have been iffy depending on how people feel about the first day of April. We have two speakers today, Keshav Singh and Rajdeep Khangura. This is not probably who you originally thought was going to be here when you first signed up for that. So it turns out that Brian, who was going to be one of our original speakers, has had a family emergency. So we'll work him into our schedule again later. And we are lucky to have Raj, who is a great person to include. Just saw a seminar from him at the Maize Genetics Conference. So the Plant Phenomics Fridays seminar series, any of you that have not been here before, this has been going on now for 3 or 4 years off and on. And we try to do a couple of people every time we get together, leave enough time for people to discuss when they have good questions, interesting ideas, or maybe even our speakers want to have a little bit of conversation. We found ways to support the Plant Phenomics Fridays over time, lots of different ways. Right now, we're funded by HIPS, which is High Intensity Phenotyping Sites, a multi-scale, multimodal sensing and sense-making cyber ecosystem for genomes to fields. That's funded by USDA NIFA. We also have funding for this series from IRA, the AI Institute for Resilient Agriculture, which is funded by USDA NIFA and the NSF. So with that, I'm going to introduce Dr. Keshav Singh. Dr. Singh is a research scientist at the Agriculture and Agri-Food Canada, specializing in remote sensing and plant phenomics. He has a PhD in hyperspectral remote sensing and a master's in engineering physics. He began his scientific career working as a postdoctoral fellow at UC Davis and went on to the University of Saskatchewan in Canada. In the last 10 years, he's been working on proximal and drone-based high-throughput aerial imaging and other aspects of digital agriculture to study various crops, including wheat, canola, and lentils. His current research involves leading the development of plant phenotyping methods using smart sensor-based systems in controlled and field environments. He's also involved in spectral imaging technology, agronomic data processing, image cube analysis, AI algorithms development, crop mapping, and big data analytics to monitor plant health. With that, I'm going to turn it over to Keshav. His seminar is entitled Digital Imaging Technology and Plant Phenotyping, Canadian Crops Perspective.
Thank you, Caroline, for this invitation. And I will spend like around 20 minutes next for working on my seminar, which is the digital imaging technology and plant phenotyping. Basically, I will address mostly the crops, including the different case studies and Canadian crop perspective. So I just want to start with three things. First, I want to acknowledge the First Nation contribution made on this country in Canada. Like indigenous people have did a lot of contribution for this country, so I have a quite big respect for them. And the second is I want to acknowledge this Einstein photoelectric effect, which is created the basic of electromagnetic radiation for the remote sensing. And this is whatever we are working, and we are using the term remote sensing. Started with the photoelectric effect, and he won the Nobel Prize in 1921 for his work. And the third, I want to do my respect for University of Lethbridge collaboration, which is the one of the very old mature group of remote sensing in Canada and serve a lot in terms of Canadian agriculture. So this is my associated research group who's contributed in different case studies, and I'm collaborating with the University of Saskatchewan. My previous work was with them, and currently I'm collaborating with different scientists in Agriculture Agri-Food Canada and different professors at the University of Lethbridge. And I respect all the funding agencies who give the fund for my different projects. Okay, so I have structured my seminar in a different points, starting from Canadian farm industry challenges and the strategies of high throughput phenotyping, how the plant photon plant interaction works and what is the different image based phenotyping platform and different imaging sensors, and how we will do the different sensor measurable parameter, which we call as a phenotypic traits, and what is the exact definition of difference from the different sensor when we talk about the multispectral imaging, and what are the different data integration and analysis workflow. So I will go through this point and I will support with different case studies, work in the canola, lentils, and wheat, spring wheat um, crop. So here are some of the key challenges of Canadian agriculture and breeding program. Crop adaptation to different environment and ever-changing climate is a big issue and now, and the frost damage, heat waves, hailstorm, prolonged weather, drought, silently, all is triggering the different point around the globe. And for the pest disease and the weed, the farmer have to spray their crop with the different chemicals, which increase the cost of the production and poisonous in the biosphere. So to improve the conventional crop management practices, we need to find an efficient way to combine the genome, phenome, environment, and management practices to predict the key phenotypic traits, which is the goal of crop breeding development program to improve the genotypes. So here, Canada is mainly known for three major crops. One is a canola, wheat, and lentils. So canola is playing a big economic role in Canadian economy and having some physical issues like swathing, pot shatter, and oil seed quality. Also suffering from the different disease, clubroot, scotonia, steam rot, and there is a frost damage during the emergence time. Wheat is one of the cultivated staple crop around the globe and having some physical issues like stay green potential and how one can do the yield prediction before time. And there's a different diseases associated with wheat like Cyprus, FHP, and leaf spot. And for the lentils, which is now one of the big plant-based protein source, also having the pod maturity issues and deciding the right time of the desiccant or spray timing of the herbicides, and having a series of different disease and or tackling the different weed problem at early stage of the lentils. So why we need the how to plant phenotyping? All the traditional way if we see of doing the phenotyping is quite subjective, time consuming, labor intensive, and expensive. So market is a global market. We need to have automatic selection of more adaptable crop varieties in ever changing climate. So the lack of adequate phenomics data is in the plant breeding program, we need to set some of the non-destructive analysis plant phenotyping techniques, and which can be addressed by different points while leveraging the digital imaging to link phenomes to the genome, which is the goal of genomic selection. So here you can see the different phenotypic traits using the any digital imaging platform, which is the germination rate, photosynthetic activities, plant growth monitoring, drought, heat, and frost damage tolerance, and plant pathogens and crop mapping. So coming to the strategies made by Plant Phenotyping and Imaging Research Center, which is the one of the biggest grant. This center is got one of the Canadian biggest guy working on plant phenotyping. They have set up the crop genetic gain strategies. So they are working parallelly into 4 themes. One is mobilizing root soil microbiome interest in genome and physiological selection of yield stability, field phenomics for breeding and precision agronomy, and using the deep learning phenomics algorithm like artificial intelligence and deep learning machine learning algorithms. So here you can we have the different sensing based platform, which is the greenhouse automatic control phenotyping, proximal field phenotyping using the ground sensing card, UV based phenotyping, aerial drones, and satellite based crop management and yield estimation models. So when we have the elite breeding germplasms or new genetic diversity, it is combined with all these 4 themes, one can have achieved the goal of deep plant phenomics or climate resilient crop under the umbrella of precision agriculture. So this is the how the system used to work when we work on the how the gene phenomics needs to be triggering the genome selection. So let's start with the basics of how the plant phenome interaction generally work to retrieve the biophysical parameter. So here is a plant. When there is a light source from the solar, it comes is a different reflective reflection take place as a different part of the plants, and some is transmitted, absorbed by the plant body and reflected back to the sensors. So plant generally reflect the green and near infrared photons and absorb all other colors, which is considered the blue and the red. So here you can see the typical hyperspectral profile, the wavelength x-axis and the reflected values on the y-axis. So comparing the stress versus the healthy plant, the stress plants, it reflects more in the green light and less in the infrared as compared to the healthy plant. So here in the basic terminology of the leaf, when all the different component of the light reflect back at the different portion of the leaf, some is reflected back toward the sensor and get absorbed, and some is absorbed by the leaf body. So by using the biochemical and biophysical parameters, there is an example of Procell model, one can retrieve all those basic pigments associated with the leaf structure. So here is the plant, some of those phenotyping platforms starting from the University of Saskatchewan, and also some is also in my group and Agriculture Agri-Food Canada in Lethbridge. So here is a Phenovan, which is the on-the-go charging station of the batteries for the drones or the UAV. We can have the flights on the very remote fields and can do the pre-processing, analyze the data on the ground. And these are the different payload sensor using M600. You can see having range from RGB, multispectral, and hyperspectral and thermal sensor on all different platform as per the requirement of the trade-based objectives. And the third is a mobile PhenoCard having series of different sensors running on, is an autonomous unit having the controller and triggering the RTK-based GPS station along with working of the base station on the nearby field. And there is a couple of indoor imaging sensor. One is a gantry-based system having high-end DSLR camera with a multispectral sensor. And my group is also currently working on Raspberry Pi development for indoor plant phenotyping. So here's some of the sensor. I'm just quickly going through the Raspberry Pi, and this is the wireless-based Firefly system is developed by the Lean company right in Calgary in north part of the Canada. And this is the Sony sensor, Mica Sen 5 band and my hyperspectral camera developed by the Corning Micro HSI, and Space ICO handheld for the indoor imaging or proximal sensing. Some of those cameras are related to the thermal is a FTLIR XT laser sensor, and Ocean Optics is a spectrometer. And this is the PhenoCard which we getting from the University of Saskatchewan, having range of different sensor. You can see ranging from thermal, depth camera, spectrometer, lidar, and all those near infrared to RGB and ultrasonic sensor. So by using all these technology, just want to convey here using the platform and associated sensor, depending on the objective, one can choose the right sensor type. So RGB can be used for canopy cover, leaf germination rate, flower intensity, and a lot of different other tests and leaf area index. Bringing the multispectral and hyperspectral, it can be used for plant health, vegetation, different vegetation indices, retrieval and phenotypic prediction, biochemical composition, photosynthetic rate, pest and insect attacks in the different type of the crops. Bringing the infrared and thermal sensor value, it can be widely used for water stress, surface heat emission, drought tolerance, and heat wave estimation in the different crops in the climate change conditions. And LiDAR is basically made for surface elevation model, like accurate having like high penetration depth, plant growth estimation, and canopy volume and structures. Bringing the radar value, like different satellites, radar satellite is widely used for soil map, soil moisture, water potential, and now the ground pattern data is also booming to bring the detection of roots in the soil. So okay, so what is the exact definition of hyperspectral imaging? So hyperspectral imaging is a combination of spectroscopy and imaging from a distance object. Here you can see the hyperspectral imaging profile, a hypercube having X and Y as a spatial axis, and lambda is as a Z is a spectral axis. And you can see a one plot hypercube. So it is coming with numerous different information as compared to the multispectral, because all the band is continuous and bringing the lot of subtle information when we do want to play with a different vegetation index or want to develop the new one, as compared to multispectral is giving us limitation, having only the handful of information and vegetation indices. So there is always an advantage of having the hyperspectral over the multispectral and RGB sensor systems. So here is my hyperspectral imaging tool or the system. You can see in the field how we are doing different measurements. Here's the hyperspectral camera. And along when we are doing the measurement in the field, we have two other components. One is a weather station, and there is a spectroradiometer for continuous tracking of the solar cells geometry to correct the hyperspectral data in the real time. And this hyperspectral sensor have 150 bands ranging from 400 to 1000 nanometer with a spectral resolution of 2 to 4 nanometer and spatial 1 centimeter, 15 meter height. So here the protocols related to data integration and workflow, I have divided into two different parts. One is a data processing and data analysis. During the data processing, we are getting the hyperspectral data from the hyperspectral sensor, passing through the radiometric correction, doing a bunch of georeferencing like orthomosaic and point segmentation, and they are going through the spectral calibration using different tool like ground, using the quick atmospheric correction, BRDF, which is the bidirectional reflectance corrections. And finally, we have the spectral profile or reflectance matrices for further data analysis. As long as we are getting the highly calibrated spectral profiles, we need to identify the prominent band or optical wave bands, need to be using the existing vegetation indices or developing the new one on the basis of these different band information coming out from the spectral profile and the bands. And then we have the feature extraction and accuracy assessment and doing the spectral phenotype classification. So here you can see in the broad system, there is a remote and there is a different vegetation or information is coming from the remote sensing sensor and having the soil survey and climate model, compiling all those in the ArcGIS using ArcGIS software. We have the crop yield prediction model. So I just want to jump on my first case study, which was done in 2018 summertime. So these are the 56 genotype of the canola having seeded with the 3 replicate in Kearney Research Farm located in Saskatoon City in Saskatchewan province in Canada. And we have started with choosing the 5 main genotype, which is the very commercially grown here in Western Canadian region. And we have collected the different time point UAV data set from at 5 time point. Here you can see at a different seed color changes stages. One is starting from having pod as 0% moisture, 25, 40, 60, and 75. After getting all those 5 time point UAV hyperspectral data, along that we are doing the field ground sampling, collecting all those seed pods and weighing their moisture content. So I have developed a canola pod maturity index, which is the function of blue, reddish, and near infrared bands. So while modeling those with respect to the pod or seed moisture versus this maturity index, we are getting a very high correlated values. And it conveys that when the seed pod moisture is less than 30% and index value is less than 0.1, this is giving the right time of swathing or harvesting the canola in the field. And hence the final outcome of this project was these index change can attribute to the moisture and seed color change, which is highly hard to predict by visual eyes or conventional methods. And it helped the breeders to make the selection of different genotype based on their maturity timing. And it can be used to determine the proper timing or spraying the desiccant. So this study is recently published in Canadian Journal of Remote Sensing. So jumping to second, so second case study was along the previous study in 2018. It was nitrogen efficiency among the different canola genotype. For that, we have seeded 4 varieties of canola genotype in the 3 replicates. That was NAM 017 and hybrid 2 hybrid varieties with 4 different nitrogen doses as a treatment. So here you can see when there is a fertilizer nitrogen applied on the soil, how it goes to the different part of the plants and goes to the atmosphere and creating the greenhouse gas emission, especially the nitrogen oxide. So we need to optimize the uses of fertilizers while we are planning and what is the right time of applying those fertilizers. So here you can see the average reflectance profile coming from these data set. I'm just representing here the zero, like having the control without nitrogen and having 150 of the nitrogen applied to the field. And so I have used the very basic nitrogen refractive index to classify that image, and it come out very well, and it's validated from the ground truth data set. So it gives the farmers or the end producers the direction how and how much amount of the nitrogen they should be using and what time in the field as per the requirement of the plant base and uptake. So the third case study was about the spring wheat at different phenological stages. We have started with 15 widely used spring wheat varieties, seeded at 3 block, 3 brigade, and 16 runs. So, so I have did like hyperspectral imaging data collection as 5 main pheno stages, which is the jointing, booting, heading, flowering, and ripening stage. Along with this UV data collection, we are also doing the ground measurement, having the head moisture collection, using the leaf chlorophyll and final yield. So there was a 3 objectives set how we can do the spring wheat varietal discrimination, identify the most prominent wave band using the machine learning algorithms, and if we are able to do the yield prediction model using the 5 different phenological stages. For that, we have used artificial different like 4 machine learning algorithm, artificial neural network, support vector machine, random forest, and partial least square regression. So here you can see the flowering stage of the hyperspectral cube of the trial. So here the average reflectance profile coming from 15 those different spring wheat varieties at the flowering stage. So all the data coming from different pheno stages is got trained on the basis of the ground values, which are the leaf chlorophyll, head moisture, and yield. And after doing a lot of modeling, we have identified the prominent bands which is responsible for different traits like chlorophyll, moisture, head moisture, and yield prediction traits. And before that, we are also run the partial least square discriminant analysis to identify all the discriminant between the 15 spring wheat varieties. So as a conclusion, we have did a lot of different modeling, combining the different weight of the model at the different pheno stages, and we have identified the partial least square discrimination is a promising tool for doing the wheat varietal discrimination. Overall results is come out good in terms of the band selection methodology, and the maximum weightage is retrieved using the artificial neural networks as followed by partial least square and support vector machine and random forest. So we have got, as a conclusion, we have found that flowering stage giving us the maximum weightage of identifying the yield prediction of having the R square of 82%. So out of all the pheno stages, flowering stage is come out to be more like 50% in terms of predicting the yield prediction. So as a follow-up, another study is about the wheat striper assessments. So here you can see the disease nursery in the Saskatchewan region, and we have the different checks. And for this study, we have started with the 4 checks, starting from very resistive towards the stripers and very susceptible towards the stripers. So here you can see the average reflectance profile of those 4 checks, wavelength versus reflectance. And by using the first order derivative, we have identified what are the prominent wave bands changing along the time. And on the basis of those change wave band, we have identified from the list of existing vegetation indices to model the severity index coming from those all striperous towards those vegetation indices. So here you can see the ground truth data coming from average severity by using the visual rating versus the vegetation index coming from different vegetation indices. So after modeling versus the different vegetation index and from the ground to severity rating, we have found the leaf rust disease severity index having the maximum R square value as compared to other choose vegetation indices. So in another like final case study, we have used the lentil. We have started to identify what is the right time of the desiccation and comparing the different herbicide component in the lentil desiccation responses study using UV-based platform. So we have started with one of the red lentil variety and choose the 8 different herbicide treatment. And on the when we are doing the ground measurement, we have doing the visual rating of the desiccation and plant moisture content by collecting the the, the different plant from the different treatment plots. And we have used a UAV based hyperspectral sensor to get the data from baseline before these spray of herbicide, and after 3 days, 7 days after the treatment, 10 days, and 24 days after the treatment. So you can see all the desiccant or the herbicide we have used for this study. And by using the partial least square score, we have plotted how we have plotted the different data points. So black is here is the control without any treatment, and we have identified how the different herbicide treatment, those plots or plants is jumping from their control. So here that here's a simplified profile of different treatments of 7 herbicide and plus 1 control, and how they are behaving from their control. So black is a control, and here different data point is associated with the treatment. And we have measured the Euclidean distance to measure the jump from their control to at certain treatment at certain date after the treatment. So starting from the control, you can see there is a variation along the time up to 20, like 3rd week or 24 days after the treatment. So we have found the equivalent distance show a comparative herbicide response in dry down of the lentil faster. So ammonium nanotate, this is the new organic herbicide as compared to other you can see in the list is a chemical herbicides. So we have found this organic herbicide has the most quicker effect from starting from the day 3 of after the treatment and running all the good effect from up to the 24 days after the treatment. Glyphosate, which is the chemical based herbicide and widely used, effect start at a slowly and attain a highest at later date after the treatment. Hence, the maturity index which we have developed for in canola case also observe as a surrogate for equivalent distance. So, and a key development as a conclusion, I just want to give some very basic points. A UAV-based hyperspectral imaging has a potential for early detection of phenological stresses like biotic and abiotic stresses, pest infestation disease in different crop cultivars, which is triggering when we say the plant health measurements. Maturity index can be used as a decision tool for swathing and harvesting of the canola genotypes and make decision for seed selection in crop phenology. Digital imaging technology can be used as a to enhance the nitrogen use efficiency among canola genotypes to optimize the grain yield. Machine learning tools or algorithm has the potential in cereal crop discrimination, band selection, and yield estimation, which is the basics of plant breeding programs. And spectral imaging are used as a study to study the desiccant response in the lentils to improve pre-harvest herbicide application to optimize the spray timing and the cost on the spray. So I just want to share one of my groups, all the plots which we have when we are doing the field work. Yeah, thank you everyone for the seminar, and thank you. I'm welcome for any question if the audience have. Thank you.
Thank you, Keshav. There was clapping in the room here, though you probably could not hear it.
Yes.
So we're open for questions. You have one in our chat from Raj. It says, can you extract data on a per plant or per inch basis from each plot with hyperspectral data?
Yes, so the goal was here, like one plot is considered as a one cultivar, like having no mixing out of the seeds. So we are just taking the average of per plot, but there is a pixelate data. Like when I'm training the model, you saw in the machine learning, I'm dividing my plot in thousands of different grids. So I'm extracting the hyperspectral data from those 1,000 grids and training that on the basis of ground sampling.
So I have maybe a related question. I was looking at your aerial images, and these look nothing like the plots that I'm accustomed to in Iowa, because we don't really keep around those plots as exposed to the soil as you do. In fact, one of the things that we do is to plant small grains in the corridors when we're growing maize. So to what degree do you think it's important for what you're doing to have those really distinctive differences between where you're growing your crop and what is the area around it, how it's cultivated?
Oh, so those are very typical breeding plots, which I was showing even in the video, and which are separated from one type to another type, having a couple of replicates around those fields. And we are doing the automatic extraction and taking all the values of either we are working on multispectral, hyperspectral on basis of what trait we want to achieve. So if I'm getting your question, like you are not growing on the same or like nicely plots.
Right.
Oh,
So
okay.
at least with maize, very often we grow some other plant to keep the weeds down rather than just having bare soil across the entire field. And so I was just wondering about how hard it would be to segment out different areas in that kind of a situation.
Yeah, so in terms of hardness, like I'm getting, if you are considering the soil background, I am applying the soil removal like techniques, or there's a different indices which we can use and do the soil removal, and it's coming out way better when we are working on hyperspectral as compared to multi or RGB, because there is a no lot of indices which you can use and remove all the soil background. But high spectral, there is a quite easy, I can say, in terms of soil removal or the background noise,
Sure.
and whatever is coming out is just the plant pixel at the end.
Thank you.
I was asking about the second case study you did on nitrogen use efficiency and then like performance of the crop and all that. So I wanted to see, like, did you see any correlation between the nitrogen use efficiency and then also yield in canola and all that? That's what I wanted to ask.
Yeah, so we started, I was just showing because of the time constraint, only one very quickly what we have used as a nitrogen index. And we have classified, because we know where we are applying the amount of nitrogen dose as per starting of the experiment. And it was very correlated when we have classified that on the basis of nitrogen index. So yes, it was correlated well, because at the end, when you are applying the different nitrogen doses, not very discrete one, from 0 to 150, if there is a very narrow range from, I can say, the zero control and having 10%, 15 or dose, on the basis of hyperspectral classification or using those indices, it is well classified results. And at the end, on the basis of those at a different time point or different stages of the crop, one can know where there is a need of putting more nitrogen and where there is a no need of applying the nitrogen. So that's quite promising, we have found.
Let's thank Keshav Singh again. Raj earned a BS in agriculture, majoring in plant breeding and genetics from Punjab Agricultural University in 2010. He went on to receive a master's degree in plant breeding and genetics, working on assessing genetic diversity in diploid and tetraploid species of cotton using microsatellite markers. In 2018, Raj earned a PhD at Purdue, working with Guri Johal in the Department of Botany and Plant Pathology, studying photosynthetic traits in maize. He's currently working as a postdoc at Purdue with Brian Dilks in the Department of Biochemistry. As a postdoc, Raj focuses on identifying genes underlying hypersensitive response and architectural traits in maize and sorghum using statistical genetics and omics approaches. For this work, he received a USDA postdoctoral fellowship for 2 years. In his spare time, Raj likes to do cross-country running. He also likes to go for long walks with his little Yorkie named Sony. Sony means beautiful in the Punjabi and Hindi languages. With that, I turn it over to Raj.
Thanks, Carolyn. Okay, so yes, I'm a postdoc in Dilks Lab here at Purdue. So I'm going to talk a little bit about some of the work that we are doing with natural variation. So genetic background are known to affect mutant phenotypes, and the work that I'm going to talk about is primarily focused on maize. In the Dilks Lab, we are primarily focusing on identifying genes underlying plant metabolism and development. And we have like specific projects that are focused on working on lesion progression using cell death mutants that I'll talk about briefly. But we're also looking at some architectural traits, for example, plant height. So I won't really focus on this, but I will talk a little bit about the lesion mutants. So a lot of these projects are heavily relied on omics approaches. So we use transcriptome data. We do quite a bit of untargeted metabolite analysis and also whole genome sequencing to gain more insight into learning more about these traits and figuring out the genes. So one of the common theme that runs across all of these experiments in the lab is we utilize natural variation that exists in the germplasm to identify any additional loci or genes that affect these mutant phenotypes. So just to give an example of how natural variation can impact a mutant phenotype, here's the data from 1988 from Scott Pothig's lab, where he shows these 3 different mutants in maize. So these are TeoPod 1, TeoPod 2, and TeoPod 3. So these are phase change mutants in 2 different backgrounds. So if you look over here, here's the wild type plant. So this is the wild type Ohio 51A inbred line of maize. And here's the Mo17. But these 2 mutants are very different in these 2 different backgrounds. So here in this background, the phenotype is really severe. You see excessive branching of these plants, but it's completely suppressed in this background. And we see also a difference in the reproductive structures. So here's the tassel and the ear. So you can see there's a huge difference in these 2 backgrounds. So the conventional approach to map some of these modifiers have been really by using F2 population. So I'm going to focus mostly on the dominant mutants. So dominant mutants are the one that you can see in heterozygous condition. So here I'm using an example of a dwarf mutant in maize. So it's a semi-dwarf dominant mutant in heterozygous condition. So if you are interested in mapping a modifier of this mutant, you can cross it to any inbred line, and you can make an F1 population. In the F1 generation, you can submit these 2 individuals to make an F2 population. So you don't want to self this individual, because if you self it, you'll actually get 2 doses of the mutant allele in the F2, and that will complicate your phenotyping. So instead, you can submit these 2 individuals so that your F2 population is segregating for heterozygous allele in the background. So when you grow these F2 populations, you can identify severe mutants in this population and also look for more suppressed mutants. And you can actually use this, these plants to do your bulk stacking analysis or some kind of mapping. So one of the problems with using F2 population is it requires genotyping of each individual, or you have to at least make bulks and do some kind of sequencing. The other problem with using F2 population is if you have a suppressor in this background that really suppresses the mutant phenotype, you cannot tell the difference between the suppressor and a wild type plant. Because in an F2 population, you have tons of transgressive variation that's segregating in the background. The other complication with this design is the environmental variation can create a lot of noise that can interfere with your phenotyping. So you can miscategorize a mutant as a severe when it was actually not severe, and it was only severe because of some environmental noise. So one of the advantages of this approach is you can actually map recessive modifiers. So the alternative approach that we are using in the lab is to make F1 populations and use F1 populations for mapping. So the way this works is you can actually cross your mutant, a same mutant, with two different lines. So this is the example here where I'm crossing with inbred A and inbred B. And you can make these F1 families. So because the F1 population or F1 family is completely heterozygous and is isogenic, so all of your wild type and the mutant plants will be isogenic to each other. So the only difference between these two plants will be the mutant allele. Moreover, because your material is segregating 1:1, you'll get multiple readings of the same mutant within the same plot, and they should all look very similar because they are genotypically similar to each other. Again, if you cross it to a different line, you can actually get a very severe mutant phenotype in another background that actually has an enhancer hiding in this background that can enhance this phenotype. So using this approach, you can actually do GWAS analysis because you can cross your mutant to a diverse panel and perform GWAS. You can also cross it to a real population and do QTL mapping. So this would be a typical example of biparental mapping. So the other advantage is you do not need any additional genotyping because you can actually take the genotypic data from your inbred line and use the phenotypes from your F1 and do the mapping because everything in the F1 is heterozygous with respect to your inbred line. So the phenotyping using F1 population is really reliable because you have multiple isogenic siblings. So you get the multiple data points from the same genotype over and over in one family. And the other advantage is the suppressors will be really clearly visible in these families because if you have a suppressor that only reduces height by 6 inches, you will see that consistently in a family that has multiple plants. So one of the disadvantages of this approach is you cannot detect recessive modifiers. And the reason is because everything is heterozygous condition to your inbred parent that you're, to the inbred line that you're using as a tester. The other disadvantage is if you're doing GWAS, if your modifier has a low frequency in the population, you will not detect that modifier. So, here's just to give you an overview on how this works is I've used an example here where I'm crossing a tester, and this is just to show 2 chromosomes of maize. So this could be any chromosome. And let's say in this case, this is the mutant allele, and because it's heterozygous, only one allele is mutant. You can cross this tester to multiple lines of an association panel or a real population and make these F1 families. And each F1 family will be isogenic, and they'll segregate for wild type and the mutant siblings. So because you have this isogenic comparison, you can actually control for the background that you are looking at. And you can compare these families across the whole population and do your mapping experiments. So then you can simply measure phenotypes in the wild type and the mutant siblings in the entire population and do GWAS using the genotypic data from these individuals. So we call this as an F1 hybrid association mapping, or also PHOM. So with this, I'm going to talk about 2 projects. The first project is primarily focused on identifying a modifier of a semi-dominant chlorophyll mutant in maize. So this work was initiated as when I was a PhD student, and I have published multiple publications using this data. And I'll only use data from the first publication to kind of make a point for today's talk. And then I'll focus on another set of mutants that I'm working on right now that we call as lesion mutants, and how we are using those mutants to identify additional genes. So because I'm going to talk about chlorophyll biosynthetic mutant, so I just want to give you a quick overview of the chlorophyll pathway. So it starts with glutamyl tRNA, And this metabolite gets converted into porphyrin through series of enzymatic reactions. And this compound can either be converted into chlorophyll using this magnesium chelatase. So this is the first committed step that performs this conversion of protoporphyrin 9 into chlorophyll. If you look inside this reaction, so magnesium chelatase is a multimeric enzyme. It consists of 3 subunits that come together and make a functional enzyme. So these 3 subunits are subunit I, D, and H. So this enzyme complex basically incorporates a magnesium ion into the middle of this molecule and creates this magnesium protoporphyrin 9 molecule. So the reason I brought up magnesium chelatase is because I'm going to focus on a mutant in the subunit I for this talk. So the reason this subunit I is important is there's already a mutant in maize that was characterized a long time ago. And this mutant is called OLYELLO1. And the allele that's mutated at this locus is the OLYELLO1 N1989 allele. So this is a semi-dominant mutant allele, and it carries a missense mutation from leucine to phenylalanine at position 176. So here's a picture of this mutant. So here's the wild type on the right side. The middle plant over here is the heterozygous plant. So you can see that this plant is slightly pale. And if you have a homozygous mutant, it's completely devoid of chlorophyll. So these seedlings die a few weeks after germination. So, but these plants over here, they grow to maturity, and you can actually perform crosses and do experiments with them. So this particular mutant allele is deleterious, as shown over here. And the way it causes this a reduction in chlorophyll is by actually competing with the normal protein to form this complex. So it competes with the normal protein, and every time this subunit gets incorporated, it actually renders the complex non-functional. So we have been working on this mutant in the lab for a few years. And one thing we noticed is that this mutant, the phenotype gets impacted by the genetic background, just like what I showed you at the beginning of the talk. So here's an example of this mutant in 4 different backgrounds. So we have a mutant in B73 background, and we crossed it to these 4 different lines. So these are F1 plots. And you can see that the white lines over here are the wild type siblings segregating in these plots. And the blue lines are, or the blue arrows are pointing to the mutant plants. So if you look at panel A and panel D, you can see that the mutants are really, really severe. They're very yellow. But if you look at panel B and C, the mutants are a lot more suppressed. They look a lot more like the wild type siblings. So we decided to carry out a foam experiment to map the modifier using this mutant, because they clearly show a great difference in chlorophyll content. So the design is just like what I showed you earlier. So in this case, we used this OLYELLO1 N1989 heterozygote plants in B73 as a tester, and we crossed it to a panel of 378 lines, and we did association mapping. So the phenotypes that we measured is chlorophyll. So we measured chlorophyll at 2 different time points, and we also measured a couple other traits in this population. So now, because we had hundreds of these families and we planted multiple replicates, so we had close to thousands of plants in which we wanted to measure chlorophyll, we decided to use a quick, rapid way to measure chlorophyll in the field. So in order to do that, we use this instrument called CCM 200+. It's actually a very simple device, and it only measures chlorophyll content from a very small area of the leaf, 71 millimeters square. And the way it works is it basically measures transmittance through the leaf at 2 different wavelengths. So it measures wavelength at, it measures transmittance in the red light and also in the infrared light. So the infrared light allows you to control for the thickness of the leaf, whereas the red light gives you the reading of how much chlorophyll the leaf has. So based on this calculation over here, you can calculate a relative amount of greenness that's present in the leaf. So we use this instrument in the population. So we also did a small experiment before we deployed it in the field, where we actually showed that this instrument does a pretty good job at predicting the chlorophyll level that we normally get from a destructive method using spectrophotometer. So the reason we didn't want to use a destructive method is because it's really slow. So we didn't want to process all these samples through this laborious method. So we decided to use the CCM to measure chlorophyll content. So we basically deployed this instrument on the F1 population that I showed you earlier, and we performed association mapping. So in the association mapping, we identified a major QTL on chromosome 10. So here's the data from that. So the x-axis over here are 10 chromosomes of maize, and the y-axis is the negative log 10 of the p-value. And each dot over here is a SNP present across the genome. And higher the SNP is on the axis, more associated this SNP is to the trait that we're looking at. And different colors here show different traits. So remember, I showed you that we measured a bunch of different traits. But what I want you to really focus on here is this sky blue dot right here. And this is the CCM that we measured in the mutants. So you see this, this CCM shows a pretty strong significant association on this region of chromosome 10. And we call this QTL-Way1. We also found other traits such as flowering time. DTS and DTA are flowering time traits here. So this is phase 2 enthesis and phase 2 silking. And MT is just the mutant. So we saw other traits as well that also showed a pretty strong association with this QTL. We also found some other minor QTLs that have been previously described to be associated with flowering time and plant height. But we were wondering what's going on with this QTL. When we looked inside this QTL, all the SNPs that were associated, they were within the 5' and the 3' UTR of the OLyellow1 gene itself. So remember, we are working on a mutant that has a mutation at this particular gene. And we found a modifier that maps back to the gene itself. So we were wondering what's going on. So we did a couple experiments, and we found that the expression of the OLyellow1 gene itself can explain the change in the CCM of the mutant. So here is the correlation of the expression of OLyellow1 gene in the inbred line. And this is the wild type CCM in the F1 hybrid. So you see there's no correlation between the expression of OLyellow1 gene and the CCM that we observe in the wild type plants. But if we look at the OLyellow1 expression with respect to the mutant chlorophyll, we see a pretty strong positive correlation, which means that the lines that have higher expression of OLyellow1 gene show higher chlorophyll content. So we characterized this QTL in this paper and basically showed that an expression change that exists in the wild type maize germplasm can explain this QTL that impacts the chlorophyll content of this particular mutant. So, so with this, I'm going to move on to the second part of the talk, where I'm going to talk a little bit about the lesion mutants of maize. So lesion mutant is a class of mutants that undergo cell death in the absence of any pathogen. So here are 3 examples of different lesion mutants in maize. So lethal leaf spot 1 is a mutation in the chlorophyll breakdown. So these mutants accumulate a lot of chlorophyll catabolites that are toxic to the cell and cause cell death. Another mutant here, LES22, is a block in porphyrin biosynthesis. So this is the enzymatic step before chlorophyll biosynthesis. So again, in this case, if you block that pathway, it accumulates a lot of phototoxic metabolites that cause cell death. And RP1D21 over here is a disease signaling mutant. So in maize, there are over 30 mutants that belongs to this category. And over 20 of them are encoded by either a dominant or a semi-dominant allele. So in this case, these 2 mutants over here are encoded by semi-dominant allele. So just like these mutants, there's another lesion mutant in maize that we call Belaflep1. So here's a picture of this mutant. You can see the wild type leaves here. There's a young wild type leaf and a young old leaf. Wild type old leaf. And here we have a young and old leaf from the Belfleck-1 mutant. So in this mutant, lesions appear in the emerging leaves and they do not get severe with age. So here you can see that the lesions are really severe on the young leaf. But if you look at older leaf, these lesions actually do not become more severe and they just stay as it is. And because this mutant stays pretty healthy and green at maturity, we call it as Belfleck. It's a semi-dominant mutant, so the homozygotes are more severe than heterozygotes. So this mutant allele is encoded by a missense mutation in a glutamate receptor gene. So we did a similar experiment with this mutant where we tried to map this modifier using the foam approach. So again, we took the mutant and we crossed it to the association panel and we did association mapping with it. So before I show you the results, I just want to show you the variation we see in the lesion severity of this mutant in the F1 population. So here I'm showing you 5 different leaves from the mutant, and you can see that the lesion severity varies quite a bit. So we actually scored the lesion severity in this mutant from 0 to 5, with 0 being no lesions and 5 being very severe lesions. On top of measuring lesion severity, we also measured chlorophyll content in these mutants using the same approach as I showed you before using the CCM meter. So we did association mapping using the lesion score. So this is association mapping data only using the scoring data. So again, on the x-axis, we have 10 chromosomes of maize. The y-axis is the p-value. And the red line over here is the arbitrary threshold. And we actually identified a strong association on chromosome 2 that we call as flectone 1. So this is the QTL name. If you look inside this QTL, we actually identified a pretty significant association at a gene that encodes for a glutamate receptor. So remember, the Belfleck-1 reference allele that we used is also a glutamate receptor, but that's actually a different glutamate receptor that belongs to clade 2. So we actually identified another glutamate receptor that modifies the phenotype of this mutant, and this glutamate receptor belongs to a different clade. So because we also measured CCM in this mutant, we actually also used that data for mapping. So one thing that was pretty clear to us was that the lesion severity and chlorophyll were negatively correlated, which is expected because if you have more lesions, you expect less chlorophyll. So here is the correlation of CCM with 2 different scores that we took on July 1st and July 22nd. So we see a pretty strong negative correlation with both of these scorings and CCM. But when we actually did the mapping, we didn't find any significant association using the CCM data. It was kind of a bummer, but when we were thinking more about it, we realized that one of the reasons this is happening is because the CCM meter that we are using is only taking chlorophyll content from a very small area on the leaf. And because it's a lesion mutant, depending upon where the lesions are, you can really change the phenotype quite a bit and not really capture what you see on the macro level that a human eyes can see when you actually score that plot. So the reason we are really interested in quantifying some of this variation using CCM or other imaging tools is because there are tons of lesion mutants in maize, and we have been developing these populations with all of them for last couple of years. And one of the setback that we often see with these mutants is scoring lesion phenotype just as arbitrary scores is quite raw, and you don't really capture the complete set of variation when you're just giving them a simple score. So we are really interested in identifying or using tools that can actually capture more of this variation using more imaging-based resources and tools. So with this, I just want to kind of end on this note. So we're thinking of switching to a higher throughput phenotyping platform. The reason I call it higher is because even though CCM is a high throughput way of quantifying chlorophyll, it doesn't really solve the problem that we have with these lesion mimic mutants. So we're not really able to capture the genetic variation we actually, or the phenotypic variation that we see when we score them. So the other problem that we have with lesion scoring is it's really hard to train people to do this. And a human eye is actually very limited at quantifying it over a broader range, because it's easy to score something as 0 and 1, but it's really hard to score something as 0, 1, 2, 3, 4, 5, 6, 7, or up to 100. So you're not really quantifying the variation in a continuous fashion. So as I showed, that CCM is a great tool, but it only captures variation from a very small tissue of the leaf. So it's really bad at capturing the macro effect of lesions that we normally see in the field. So all the QTLs that I showed you that we detected as significant association, they only explain 20 to 30% of the variation that we observe in the field. So most of the variation is actually not being explained by any genotypic difference. And that's because we believe these phenotyping methods that we are using are missing out on a lot of this variation. So finally, we would like to move more to the spectral sensor data, where we can really see this quantitative difference, and that can really eliminate most of these problems. And this will allow us to detect not only just novel loci that we are not able to detect with the phenotyping approach that we are using right now, but it will also improve the precision with which we can map these loci. And moreover, this will also allow us to look at the temporal effect of these genes over time, because if you're using sensor-based platforms, you can collect data multiple times instead of like only once or twice a season. So with this, I just want to say that we are looking for imaging solutions to collect these data points. And we are really interested in collecting data from each plant if possible. And part of the reason for that is because the F1 design that we're using for mapping is the material is really segregating within each plot. So we would really like to collect data from each plant. So last year, we actually did collect RGB and hyperspectral data using drones, but we don't have capacity as of now to extract information from per plant basis. So we are really looking for collaborations in this space to get most out of these experiments that we're doing. So, foam is a powerful design to identify these interacting loci. And it's really good at identifying major QTLs, but we really think if we can deploy some of these sensor-based approaches, we can extract a lot more genetic information. So with this, I would like to acknowledge my mentor, Dr. Brian Dilks, and members of the lab, and the research and the core facilities at Purdue, and the collaborators on some of the projects that I talked about today, and also the funding for all these projects. With this, I'll be happy to answer any questions. Thank you.
Thank you, Raj. So the first thing I will do is to alert you, if you're not paying attention to time, that it is about 5 minutes after the top of the hour. So if you need to jet away, don't feel bad about zipping out of the room or out of the Zoom. However, if you want to spend a couple of questions, a couple of minutes on a couple of questions, feel free to stick around. The first question is in our chat. Has foam been tested on traits which show up only with 2 or more genes being mutated, or better with lines which differ from major QTLs, but not essentially coming from mutated populations?
Yeah, I'm still processing the question. So most of the traits that we have looked at so far, so all of the testers that we have used are monogenic mutants, right? So these are mutations in single genes, and for all of them, so far, we have only identified up to 2 to 3 QTLs. And in every case, there's a major QTL and maybe a couple of minor QTLs. And I'm assuming if you actually use a real population, you have a lot more power to do to detect these QTLs. We have only done biparental cross with one mutant, and that's all yellow. In all yellow, there is only one QTL that impacts chlorophyll. So my assumption is if you actually use real population for more of these mutants, you might be able to detect more QTLs because your allele frequency is really high. So you have a lot more power to do the analysis. But with GWAS, I'm not sure if you'll be able to do that as successfully.
Thank you. Any other questions for Raj? If not, let's thank him again. Raj, I especially thank you for showing up with a great seminar on short notice. Thank you both, Keshav and Raj, for joining us for Plant Phenomics Fridays. Next week, we have Roland Schaffleitner and Stephanie Green. They'll be talking about germplasm and gene bank. So if those are topics that you'd like to join, we look forward to seeing you.